So, in the last video, we looked at the function of the rough and smooth endoplasmic reticulum, and we looked at projections, and we reviewed the other organelles as well. Now, if this was an epithelial cell, it's going to have little tiny cilia on them. And what do cilia do? Cilia move things. And what happens is they move things, and so most viruses and bacteria that get into your respiratory tract cannot make contact with the cell, and they get washed away. Now this guy's the coronavirus. So see the spiky proteins? Those are the grappling hooks. And they let it grab onto the cell. And that's actually how it puts its RNA in. So the coronavirus has a grappling hook. Now the whole job of a virus is to get it to be reproduced. So what happens is this RNA actually codes for all the viral proteins. So the virus is basically lipids and proteins, and it's going to leave the cell. So what happens is this RNA goes to ribosomes to be made into more proteins. Now this is going to be proteins that leave the cell. Will it be free ribosomes or on the rough ER? That's right. It goes to the rough ER because the rough ER makes proteins that leave cells. The protein needs to be then packaged in its vesicle. So that's going to go to the Golgi complex because that packages proteins. So who would have thought I can use a virus to teach protein cellular structures in animal cells? So this lecture is on mitosis. And we're going to look at the phases of mitosis. And you're going to be able to identify the phases in lab. So there's two phases of the cell's life cycle. There's interphase, which it's doing normal cellular activities. Now it could be preparing for cellular division. Some cells are in interphase for a very, very long time. Some indefinitely. Muscle cells do not divide. They're always going to be in interphase. Now, cells that divide a lot, like epithelial cells, they go through mitosis, where the cell divides and gets replaced. In the meiotic phase, the cell is going to divide into two identical cells. And there's 4 stages: prophase, metaphase, anaphase, and telophase. What do any of these words mean? Pro means first. But what does telo mean? Well, what does telo mean? Telephone, television. It means across. Does anyone know what meta means? It actually means middle. And if I'm remembering correctly, ana is apart. So let's look at our cell. This is Bob. He's a cell. And he's an interphase. So I could see a clear nucleolus in the nucleus, but I can't really see the chromosomes. Now, during this time, the chromosomes double in number. So you have 2 sets of each chromosome. When the cell starts to divide, the chromosomes, that's where the DNA is, become visible. So this cell is going to have 2 chromosomes. And what also happens is the nuclear envelope vanishes. And the centrioles migrate to opposite poles. So again, in prophase, chromosomes become visible, envelope disappears, centrioles go to opposite sides. So here are the centrioles. Then we have metaphase. The chromosomes are going to line up across the center. So here's our blue chromosome, pink. Now spindle fibers are going to go out from the centrioles and connect. That's the centrioles' job. They make these spindle fibers. So what it's getting ready for is a giant tug of war. This centriole is going to pull. This centriole is going to pull. And the chromosomes are being held together by these little centromeres right here. What do you think is going to happen? Yes, like my 2 kids fighting over something, they're going to rip it in half. I have no idea what happened to my cell shape right here, but in anaphase, the chromosomes are pulled apart. Now, this is the shortest of the phase. It's the hardest one to find, so if we actually were in lab today, you would have to find this on these fish eggs, and it would be very, very hard to find. I actually did a project for a stats class where I counted the percent of cells in each phase. Anaphase was the worst phase to find. Now, the last phase is telophase. I like combining this with cytokinesis. The chromosomes have now migrated to opposite sides, and the nuclear envelope reforms. Now we get cytokinesis at the end of telophase. Cytokinesis is basically dividing everything in equal halves. So each half of the cell, this is going to become two cells, gets half of the organelles, half the cytoplasm, etc. So cytokinesis, the cytoplasm is split up. Now normally I draw the cells vertically. This is very scary to draw on the internet. So what happens is we get this thing called a cleavage furrow. And the cleavage furrow is going to start to have these invaginations, or these crevices. And right now he looks like a very sad praying mantis or space alien. But this furrow is going to start getting bigger and bigger and go all the way down. And I successfully drew a space alien, I think, or a very angry praying mantis. But this invagination is called a cleavage furrow. Now you see why I was worried about what I was drawing. So in reality, this cell is going to be more round, and then this cell is going to be more round. So we end up with two cells that are identical. So in our prophase, we have our chromosomes, and they're actually double-stranded, so they're showing them double-stranded here. And they become visible. The centrioles appear, and the nuclear envelope vanishes. In metaphase, see how they are all nice and across the middle. This is my favorite phase of mitosis. I think because it's the easiest one to find, they put this phase on all the pictures too. So the chromosomes are lining up across the middle. Spindle fibers reach out to attach to these centromeres in the middle. So the centromeres are going to grow out. The spindle fibers grow out and attach to the centromeres. And they're getting ready for a ginormous tug of war. Anaphase, see, they're getting split apart. They're getting pulled to the opposite sides. So these are going that way, these are going that way. And lastly, we have telophase. In telophase, it's officially the chromosomes disappear, the nuclear envelope reforms. I like throwing cytokinesis in, it just makes everything neat. In cytokinesis, we have a pinching, I'm so not going to draw it there. We have a pinching of cells, and that is what I was afraid of drawing earlier. To help you remember, my 60-year-old high school biology teacher helped me remember the word by using the model very graphically. I'll let you use your imagination. But I was a female teacher. The cleavage furrow is the splitting. That's how you remember cleavage, unfortunately. Please don't go complain to the dean. So, on the test, you're going to have to do two things. 1, define the phase. 2, put them in order. So right now they're out of order. So anaphase, what is that? So anaphase is D. Chromosomes are pulled apart. Metaphase, my favorite phase. They line up across the center. So that's A. Prophase. Dun, 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 dun. Chromosomes appear. That's C. And then B, the nuclear envelope reforms and it cleavages into two cells. Now we're going to go into types of cells. This is an introduction. We're going to talk about these types of tissues in the next lecture. So we have nerve cells, we have muscle cells, we have epithelial cells, and we have blast cells, which are mainly in connective tissues. So where do we have nerve cells? That's right, the brain. Where else do we have nerve cells? Nerves. And what would some characteristics of these be? What do they have to do? Send and receive. Now, do you know what organ uses most of your cellular energy? That's the brain. So, would nerve cells have a lot of mitochondria or few mitochondria? Probably a lot of mitochondria. And what else about nerve cells? Do you think they divide a lot? What happens if you damage a peripheral nerve? usually you lose functionality. And peripheral nerves can grow back. Or in a stroke, where parts of the brain gets damaged, you lose functionality. So nerve cells don't really divide. So would it have a centriole? No. Muscle cells. What do muscle cells do? They contract. And do you know why some people say eat meat? Because it has what substance in it? Protein. Needs protein to contract. So meat, as well as legumes, is a good source of protein. So we're talking about human anatomy here. So muscles make a lot of protein. So what would they have a lot of? That's right, ribosomes. Epithelial cells, I've said this a few times. What would epithelial cells, what's epi mean? They're on top. And they get rubbed off a lot, so they probably have centrioles. And blast cells, what do blast cells do? I have no idea what blast cells do. So, nerve cells. They're large cells with processes. Remember we talked about cilia, which are appendages that move things. We talked about microvilli, which extend surface area. Well, these have processes that send and receive signals. They have a very prominent nucleus and nucleolus. What's the nucleolus make? Ribosomes. What do ribosomes make? Proteins. So this stain, see how they're actually very, very, very dotted in here? That's actually something called Niesel bodies. So this one actually labeled the Niesel bodies. And those are ribosomes on the rough endoplasmic reticulum. There's lots and lots of these because nerve cells make lots and lots of proteins. So they can't divide, so they'd be lacking centrioles. And they have a high energy demand, so they have lots of mitochondria. So those are characteristics of nerve cells. Now we're going to look at the tissue in our next lecture. Now, muscle cells. They can either be long fibers or short tapered cells. And I didn't talk about cell shapes, but this would be fistiform. And skeletal muscle has an extensive smooth ER. We're going to learn it has a different name. And cardiac muscle has this extensive smooth ER, but it's not as much as skeletal. What does smooth ER do? Fats, detox, and stores calcium. Well, in this case, it's going to store calcium because it's needed for contraction. And your skeletal muscle is actually lazy. When you're not active, it's using fat for energy. Now, it lacks centrioles. So you hurt your muscle, can the muscle divide just to repair itself? No. They have proteins for contraction, so ribosomes. And lots of mitochondria because they have a high energy demand. So epithelial cells, epi is upon. So they're lining things like the mouth, your skin, your digestive tract. Now, these cells are constantly falling off. All that dust in your house are actually skin cells. So they actually have to frequently divide to replace. So they have centrioles, the other two didn't. And they're going to have a free surface. What can those free surfaces have? In the respiratory tree, we have cilia. In the digestive tract, we are going to have microvilli to extend surface area. Now, because they're active secreting or absorbing things, secretion is getting rid of something like sweat. They have lots of mitochondria. And the secretion is either going to be protein, rough ER, or fat, smooth ER. And seeing how I have 30 seconds left, we will look at the connective tissue cells in our final video.